Well, welcome to our video for this week from Charles Darwin University and the Bioscience unit. This week our topic is cells. First we're going to talk about the anatomy of the cells. Now they carry out all the complex chemical activities that are needed to sustain life, and we'll touch on some of those in this unit. They're the building blocks of all living things, and they make up tissues of cells that are of a similar structure and function. We're looking at cell structure. They have three main regions. The nucleus, which contains genetic material. The cytoplasm, which contains the cell metabolic machinery, the chemical machinery, and the plasma membrane, which surrounds the cell. Looking at more detail at the nucleus, it's surrounded by an envelope which has two layers. It contains chromatin, which is the genetic material, and in the resting phase it's featureless. It doesn't have any particular visual characteristics. In the centre is the nucleolus, where ribosomes are made, and we'll talk more about ribosomes in a moment. We have nuclear pores in the envelope, and it's through these pores that genetic material is passed to the cytoplasm in the cell. And we've got what's called the rough endoplasmic reticulum, which is a continuation of the nuclear envelope, and it's where protein synthesis takes place. Now, looking at the plasma membrane, it's quite a complex structure. The main part of it is made up of what's called phospholipid molecules. These have a head which is got an electric charge, and it's made up of phospholipids, so that's phosphorus attached to a fatty substance. And they've got tails which are facing inwards, and the tails actually have no electrical charge of their own. Outside the plasma membrane is the extracellular fluid surrounding it in the tissue. Another name for this is interstitial fluid. Inside the cell and held within the plasma membrane is what's called the cytoplasm. Now there are various protein structures in the cell wall, and we'll talk about what they do in a little bit more detail as we proceed. There are various types of proteins. This one is a glycoprotein. This means a sugar group is attached to it. Some proteins have lipids, and in this case a glycolipid, which is a sugar and a lipid, or fatty substance attached to it. Cholesterol is a very important part of cell walls, and the body makes most of its own cholesterol for this purpose. And there are channels in some proteins that allow water and small molecules to pass through. Now cells have specializations. This is sort of a generic representation of them. Not all cells have all of these specializations. But looking at microvilli for a start, these are projections from the cell wall. They're very common, for example, in the small intestine, where that's where absorption of nutrients takes place. And so what happens is that the way the microvilli are structured, it greatly increases the surface area and makes absorption of nutrients more efficient. A tight junction is where you want a leak-proof seal between cells. And again, that occurs in the small intestine, where you don't want leakage of material from within the intestinal lumen through the intestine wall. So these are leak-proof seals between cells. You have desmosomes, which are anchors which hold cells together, and they're common where there's lots of wear and tear on cells, as in the skin and also in the esophagus. And in some cases we need to get transmission of messages from one cell to the next, and so gap junctions are used for that, and they occur in cardiac muscle, for example, where you want transmission of nervous impulses. Now, looking at the cytoplasm, so we have cytosol, which is the fluid that suspends all other elements. We have this metabolic machinery that I mentioned before, and we'll discuss them in more detail. And you have various inclusions, stored substances. Fat is a good example. So let's look at some of the components of a cell outside the nucleus. First of all, we've got what's called the smooth endoplasmic reticulum, the smooth ER. This has got nothing to do with protein metabolism. It's more to do with lipid metabolism and cholesterol metabolism. The cytosol we've mentioned is the liquid in which all of these elements are suspended. The lysosomes are the the garbage bins of the cell. They digest remnants of material that are no longer being used. Mitochondria are the powerhouse of the cell. They provide the energy for all of the cell's metabolism, and they produce a compound called ATP, which we'll talk about later, adenosine triphosphate. Centrioles are used in the cell division process when a cell copies itself. This is taking place all the time. We'll talk about those in this video as well. And then over here we've got the rough endoplasmic reticulum. This is the site of protein synthesis, very important function in the cell. And the little red dots that you see are ribosomes, and they are actually produced in the nucleolus. You remember I mentioned it before, and pass out through those pores. Those are the ribosomes that I mentioned, the red dots. And then proteins that are produced by the endoplasmic reticulum are packaged up in what's called the Golgi apparatus. Now, the Golgi apparatus was one of the first of these cytoplasmic structures seen under the microscope, and the man who saw them was Mr. Golgi, an Italian researcher. Peroxisome are areas where what's called free radicals. These are positively charged ions that can cause damage in the body, and they are detoxified by the peroxisome. And then we're just pointing there to intermediate filaments that are part of the internal structure or skeleton of the cell, and also to the microtubules, which are used in cell division. Now we go on to look at cell physiology, and particularly the movements of substances into and out of the cell. So from the interstitial fluid or extracellular fluid outside the cell to intracellular fluid inside the cell through the plasma membrane and in reverse. Now there's some terms that you need to be aware of about solutions. First of all, a solvent is the medium in which a solute is dissolved. Polar molecules are those with electrical charges, and permeability is the characteristic of the plasma membrane that allows some substances to pass through but not others. It's a selective attribute. Now, going back to solvent and solute, an example is if you put sugar into water, you can watch it dissolve. So the water is the solvent and sugar is the solute. And we're going to talk a bit about diffusion. There's a couple of different kinds. It's the passive transport of substances from higher to lower concentration through the cell wall, and no energy is required within the cell. So simple diffusion is usually with water or small size molecules, and they go through those pores that I mentioned before. Osmosis is where, and it's from higher to lower concentration. Osmosis is movement of water through those pores from a low concentration of a solute to a higher concentration of solute. Facilitated diffusion is where proteins in the cell wall enable diffusion across the plasma membrane. And filtration is where liquids, body liquids, are forced through membranes by blood pressure. Mostly this is, or the best example of this is in the kidneys, where blood comes in at fairly high pressure, and the liquid part of the blood is pushed into the substance of the kidney through what's called glomeruli, and then the kidney returns the substances out of what was the plasma, the filtrate, back into the blood supply, and what's not needed by the body is then excreted in urine. So that's filtration. However, there are some active processes, transport processes across plasma membranes, and the most common one is what's called a solute pump. And here we see sodium from inside, it's a sodium ion from within inside the cell being excreted to the outside of the cell, and in the process you can see there ATP, adenosine triphosphate, loses one of its phosphate groups, becomes adenosine diphosphate, and energy is released in the process, which enables this to take place. In that second and third phase, you've got potassium being taken from the extracellular fluid and returned back into the cytoplasm. Now, this process is how nerve impulses are transported through nerves. There are some other active transport processes. One is called exocytosis. This is where the cell secretes substances into the extracellular fluid. And you don't have to remember all of these stages, but just to remember that you've got a vesicle, which is containing a substance. It could be mucus, for example, or a hormone or whatever. And there's a fusion that takes place between it and the cell wall, and then whatever the vesicle contained is released into the fluid around the cell. There's another process where the cell takes substances into it. So in the first instance, we've got in perhaps a toxic substance being brought into the cell, being taken up by a lysosome, being digested, and the harmless products being released as a result into the cell cytosol, or it could be, they could actually be, after they've been detoxified, transported out of the cell by way of exocytosis, or you can get reusing of membranes and materials by the cell. There is this other process shown here on the right, where, so this is extracellular fluid in the sort of greeny blue colour, and a bacterium or a virus or some foreign substance is grabbed by the cell, engulfed, and then dealt with internally in the cell. And in a similar way, we've got receptors on the cell wall that bind with materials or substances in the extracellular fluid and get used in processes within the cell, and this is common in, for example, immunity reactions. Now we're going to go on and talk about mitosis. It's necessary because cells only have a certain lifespan, and what happens is a parent cell divides into two identical daughter cells. Now the DNA in the cell is copied so that there are two, this is the chromatin that I talked about before, so that there's two sets of DNA, so that each daughter cell ends up with identical sets. Now, as I mentioned before, DNA in the nucleus is in a thread-like form in chromatin in the nucleus. And we need to just talk about DNA for a moment. It's made up of what's called nucleotides. These are substances, chemicals made up of a phosphate group, a sugar, and then the nucleotide itself. Now you don't have to remember the chemical formulae, but do just remember the names. And the nucleotides, you've got thymine, which always goes with adenine, and you've got cytosine, which always goes with guanine. Now if you look at the DNA molecule, we then have the phosphate and the deoxyribose sugar making up these outer helices. So DNA is made up of two helices, which are held together by hydrogen bonds, which are weak bonds. That's why they're shown in as dotted lines. And then these nucleotides, which bridge across, and they always go together in that order: T with A, G with C, and that's our whole genetic structure made up in that way. So the first phase of cell division is called prophase, and in that first phase the chromatin forms into chromosomes, and they've split in two, so there's two identical copies. They're called chromatids, sister chromatids, joined together by a centromere. So that's the first phase. So when scientists first discovered chromosomes, it was only when this stage was started that they were able to see them. Prior to that, they couldn't. You weren't able to see this genetic material. The other structures important are the centrioles and the spindle. that forms, and you'll see that in this next phase where the centrioles go to the opposite poles. The nuclear envelope is busted apart, so that's and then you've got the sister chromatids being pulled towards, and we'll see in this center equator, and you can see the chromatids there, the sister chromatids, with the centromeres, and that's called the metaphase plate, the line on which they align up on. Next phase is anaphase, where they're pulled towards the centrioles, and that's in preparation for the next phase, which is telophase. And here we see a new nucleolus forming on either side, and the nuclear envelope forming to enclose the nucleolus, and the chromosomes are organized into the form that then surrounds the nucleolus, and a cleavage occurs, and the cell split, and this is called cytokinesis. Now, just go on to talk about DNA in a little bit more detail, and in particular protein synthesis. So just going back to DNA, you remember how it's formed. It's made up of nucleotides with a phosphate group and a sugar group on the outside helices, and then with the nucleotides bridging that via hydrogen bonds. And you can see this is the phase as in preparation for cell division. You can see the DNA being split down the bottom to form two separate DNA strands with a full complement of nucleotides. So here we have, in preparation for protein synthesis within the nucleus, the unzipping of part of the DNA, and the a complementary messenger RNA is made. So the nucleotides on the mRNA, the messenger RNA, complement the unzipped portion of the DNA. So the next stage is the mRNA leaves the nucleus, which through the pores. That's what I was talking about before. By the way, the stage of unzipping and making of the mRNA strand is called transcription. So now we go on to the next phase, which is where amino acids, which are the basic building blocks of proteins, are gathered together by what's called T transfer RNA and brought down to the ribosome, and they join up with the strand of messenger RNA, and from there the amino acids are added to what's called a polypeptide chain to form the new protein. And then when tRNA has finished its job, it's returned to the cytosol and is available for reuse. Okay, so that comes to the end of our video on cells.